Hello, everybody. Thank you for coming to the Uncovering the Cellular Drivers of PSC Discovery and Innovation Through Collaboration breakout session. Today, you're going to be hearing from Sonya MacParland and Mary Vyas. Dr. Sonya MacParland is a senior scientist at the Ajmera Transplant Center at the UHN and associate professor in the University of Toronto's Department of Laboratory Medicine and Pathobiology, the Department of Immunology. She holds a Canada Research Chair 2 in liver immunobiology, and Dr. MacParland's research program is focused on translating fundamental knowledge about the immune biology of the liver into clinical applications. Her team recently developed the first transcriptomic map of the human liver as a platform to examine how immune dysregulation drives liver diseases. And then as well, Mary Vyas, president of PSC Partners Canada. Mary Vyas is the president of PSC Partners Canada. She has volunteered and led the Canadian affiliate since 2015. And Mary Vyas is a parent of a PSC patient who was diagnosed as a teen. So please give it up for Mary and Dr.
Thank you, Neil. So we're here today to talk about uncovering the cellular drivers of PSC. We have here collaborators and financial support. Sonya has no conflicts to disclose. And I'm going to hand it over to Sonya.
All right, thanks, Mary. So this is going to be an interesting conversation because we're talking not just about the work that we're doing, but we're talking about how we're incorporating the patient voice and patient partners into each aspect of the research. And it's really actually been a very interesting journey for us, and it's made our research and the trainee experience very rich. So we kind of want to talk about what we're doing scientifically, but also talk about what we're doing collaboratively and how we're forming a community around this research. So there'll be a little bit of both. So as Mary mentioned, what we're really trying to do is map the liver in PSC so that we can identify some targets. And ideally, these targets would lead us to drugs that we can repurpose that are used for other autoimmune diseases, and we can start repurposing these drugs to treat PSC. So if any of you were part of the pathophysiology discussions yesterday, we really want to start feeding targets to these people that have these beautiful animal models and these beautiful cellular models, these beautiful organoid models. And so the first step to really generate these targets is really for us to understand the healthy liver. And it's kind of bizarre that we really didn't understand what the healthy liver looked like. And so we knew that the liver was very important for metabolism, for breaking down or for clearing antigens, but we really didn't understand what each of these individual cells were doing. And so that makes it very hard for us to see which cells are dysregulated in disease and how we would target them. So, and one of the things we really didn't understand as well was where these cells are with respect to the repeating units of the liver. So what you see here are these hexagonal units, and these are the repeating units of the liver. And what we really want to understand is not just what cells are in the liver in health and disease, but also where they are with respect to some of the pathology we see in primary sclerosing cholangitis. And so we're actually part of a global community that's really mapping the healthy liver as well as the diseased liver. And we're really asking questions as to what does the liver look like in fetal life, in pediatric patients, in adult patients, so that we can have rich comparators for patients along each point in their PSC journey. And so this is work that really can't be done by individual labs. It has to be done collaboratively, and this kind of started us in our collaborative journey. And this is work that we've been doing through the Human Cell Atlas as well as the Chan Zuckerberg Initiative. And so really, we wanted to start from the limitations of our understanding of just regular liver biology. So prior to 2016, 2018, we understood the liver as the average of the cell populations that are found in the liver. So we would take a biopsy and we would do a PCR, which is a polymerase chain reaction. It amplifies the signals in the whole tissue sample you get. So you kind of get a smoothie. And from a smoothie, if you have a cell population that is the largest cell population, it's going to overwhelm the taste of that smoothie. But what we're really interested in understanding is the cells that make up the smoothie. For example, in the liver, most of the cells are hepatocytes, so they can overwhelm a signature, a cellular signature. However, hepatocytes are tuned by the cells that are surrounding them, and especially cells that are coming in and out of the liver. And so we really wanted to understand the individual populations that might be tuning the microenvironment in the liver. So what we've been using is single-cell RNA sequencing, and this is a bit of a busy slide, but what this allows us to do, as opposed to taking a big piece of tissue and looking at all the signatures in that tissue, which will represent thousands of cells, we take individual cells and we amplify the signals within those individual cells. And what that allows us to do is look at rare populations that may just appear in disease, as well as look at populations that might be absent in disease. And so what I'm going to show you today is how we kind of represent some of that data, and most importantly, how with our patient voice, we've designed this study to really look at some of the early targets or the early dysfunction that we see in primary sclerosing cholangitis. So you'll see some of these maps that I'm showing up here. What we do with single-cell RNA sequencing is we cluster cells based on the signatures that are found in those cells. And when we cluster them, what happens is these individual cells form clusters. And when we see those clusters, you see the cells that are closer together will form a cluster, and cells that are further away also will form other clusters. And so it really allows us to see if, for example, a population of bananas comes up in disease, this might be the target, it might be a target that we can focus on. And if there's a population that is absent in disease, this is a population we might want to try and promote. So throughout the presentation, we'll show you a lot of plots, but really what that allows us to do is really visualize some of the targets, okay? And then the other thing I just want to really emphasize, again, because we're working as a community, and in Toronto, we have patients that may have a certain demographic background. They certainly have a different geographic background. So as a community, we really need to develop the richer the maps, the better the targets. So what we've done as a community is really up front, we've shared data, we've shared protocols, and that's allowed us to develop maps that can be integrated together. So one of the things that we're really trying to do as part of our team grants is make sure that we're sharing all this data and we're sharing our approaches so that we can integrate our data and we have the highest quality maps possible. And so for any of the data I'm going to show you today, that's completely shareable, and this is kind of how we share our protocols through protocols.io. And so using single-cell RNA sequencing, we developed a map of the healthy liver. And this is a paper that we published in 2018. And what we were able to see was that there was multiple populations within the healthy liver. And what we were most interested in were these myeloid populations or macrophage populations. So these are immune populations that go through the liver and they pick up antigen and clear antigen. So they're kind of the eaters of the liver. And what was interesting was that we saw multiple populations of these cells in the liver. And what's also interesting is if we take an animal model and we get rid of all these macrophages and we perform we cut off half the liver, so it's called a hepatectomy, that liver will not grow back as well. So it suggests that these cells in the liver are promoting regeneration, promoting normal liver replacement. And what's very interesting about these populations is that they're also able to be targeted with nanoparticles because they are the eaters of the liver. So if we find a cool target by our single-cell RNA sequencing, we can probably deliver it through these cells because we know from prior literature that they'll eat anything. And so we could deliver signals that maybe quiet the immune system or activate the immune system. And so as we started to look in liver disease, we wanted to examine the complexity of liver disease, the cell populations that are found there, and then we also wanted to use this as a platform to find new targets. And so what we found when we started to look in liver disease is that because of the scarring in the liver, some of the populations in the liver were hard to release, so they're hard to dissociate. We also found that hepatocytes, which are tuned by all these other cells, are very dissociation sensitive. So every time we manipulated them, they would die. And so we developed a workflow that we could use two different approaches. So one is single-cell RNA sequencing and one is single-nucleus RNA sequencing. So we're taking just the nuclei out of the cells so that we could bypass the need for tissue disruption or dissociation. And so we did this study to create a toolbox for people who wanted to look at the liver, a fibrotic liver, but didn't really know what approach to use. And so what we were able to show as part of this toolbox is that if we really want to look at cholangiocytes or mesenchymal stem cells and hepatic stellate cells, which are part of the disease process in PSC, we could really use single nucleus RNA sequencing. So it helped us kind of refine our healthy map to make it more applicable to disease. And then what we found is that if your question is centered around immune populations, then single cell RNA sequencing is the best approach to look at those cells because of the fact that a lot of the genes that would give you a signature to look at the immune populations are found in the cytoplasm of the cell. So the cytoplasm is discarded when you remove the nucleus of the cells. And so what we wanted to do, again, because of the fact that PSC is so patchy, so you can biopsy different regions of a patient with PSC and you see a different disease pattern, we wanted to put our signals in a spatial context so we could associate it with a scar that might be found in the liver. And so we're using a spatial transcriptomics approach to really start to pull out the transcripts and put them in their place in those repeating units of the liver. And so this is in collaboration with really great pathologists who then map out the liver. And so they map out the healthy liver. They also map out the fibrotic scars in PSC. And with our wonderful computational biologists, they can take these signals, perform something called a principal component analysis, and figure out, without really having hypotheses at all, what is the distribution of these signals within some of the landmarks in the liver. So we got to see that if you really have a question about how to modulate or target hepatocytes, using spatial transcriptomics is a really good approach. And so this led us to our disease work with PSC Partners.
So what I wanted to talk about today a little bit is our collaboration and the fact that you may have heard in my intro that I am a mom to a young adult living with PSC. There was nothing in there about being a scientist. So, what can we bring to a study of single cell projects? So I wanted to share a little bit about some of the activities that PSC Partners does that as a patient community you may not be aware of. And this leads to how this collaboration came about. So we, as you know, our mission is to drive research and provide support and education. And the two major programs that we have done have been the grants program and our research program, which you heard a little bit about today. But we also do things, and I also forgot, of course, our conferences, our support programming. But some of the things we also do to support these more indirect activities to drive our mission, we attend and exhibit at professional meetings such as the ASLD, EASL, and CASL. And we do that for various reasons. We do that to learn what's going on in the field, but we also do that so that the researchers and pharmaceutical representatives know about us, know what we can do to support their work, know about our grant programs, but also because it's a great way to meet people. And so in 2018, Rachel and I were attending the Canadian Association for the Study of Liver Disease in Montreal. And one of the researchers we know said, "Did you hear that talk just now by Sonya MacParland? She's doing very interesting work and you should talk to her." So we did. We met, we talked about her project. We told her about the patient registry, which piqued Sonya's interest in how we might be able to support some of her work. And at the time, Sonya and her team, a very large collaborative team, were working on an application to the Canadian government for funding to study PSC in the adult liver. And we were invited to become collaborators on that project and provide some input on the application and the protocol. They were awarded the grant, and we worked together, and we incredibly generously and openly, Sonya invited us to participate, Rachel and I and another patient representative, to attend monthly meetings to provide input on the protocol, et cetera. And somewhere along that line, Sonya was invited by CZR. somehow they heard about the collaboration and was invited to present to a group of single-cell scientists about the collaboration. So it was recognized externally that there was something unique going on here. And then, interestingly enough, CZI posted an RFA that kind of described a little bit about described how we were working together. This RFA was quite interesting because they were looking for patient-partnered collaborations to jointly apply for very scientific, abstract kind of studies. One was the single-cell studies in pediatric immune diseases, which we did put together an application, applied for, and now we have a new grant, and this collaboration is going to be working together to look at PSC, incorporating the patient voice in the science for the next 4 more years. So now we're going to talk about these 2 projects.
And it's quite interesting, actually, because, as we finished our studies or as we were moving from health to disease, we were actually learning about PSC while Mary and Rachel were learning about our single cell work. So it was interesting because, as we wrote the grant, we were saying, well, we'll enroll patients, they'll come and get biopsies, and, we want to have it very quick in and out. We won't want to waste any of their time. And, Mary and Rachel were kind of like, hey, if a patient is going to consent for research-only biopsy, and now we've had 8 research-only biopsies and 6 diagnostic biopsies, so biopsies in early stage of disease, they were saying if patients are going to participate in this research, they're going to want to spend the time. So that, as opposed to having a quick in and out and doing a biopsy in the clinic, put them through interventional radiology, make sure they have, the proper pain control. And actually, we had never had research-only biopsies before Mary started working with us, right? And so it really, and as we're looking towards developing those really rich targets for our animal modelers, for our organoid modelers, for people that are doing tissue on liver on a dish, the earlier stage of disease might be the most informative because it's not confounded with some of the complications of late stage disease. So yeah, and there's been a lot of situations where, as we were writing the grant, we were corrected on some of our terminology and some of the tests that we would want to have as an exclusion or inclusion criteria. So it made it very, very fun, I think, and very informative, and it kind of added another level of validity to the work we were proposing. And so we have 2 studies. It's a single cell study of adult PSC, and this is called the PSC CAN study. So this is funded by the Canadian Institute of Health Research. And this is a wonderful collaboration between the Toronto General Hospital Ajmera Transplant Center, Sick Kids Hospital, University of Toronto, as well as PSC Partners, and we have some collaborators at McGill University. And really what we want to do is, again, define and target autoimmune liver disease. And so we had as many people at the table as we could possibly recruit, including clinicians, surgeons, patient partners, bioinformaticians, clinical research coordinators, and basic scientists. And so we would have these monthly meetings where everybody would be in the room. And it's important to just keep everyone in the loop. For example, if a clinician has a patient or a patient partner has a colleague or if, someone they know that are going in, it reminds them that, this is the study we're doing, and it reminds the clinical research coordinators to recruit them. And so our patient recruitment was centered around patients with early, late-stage disease, patients that are transitioning from pediatric care to the transplant center or to adult care, as well as alternative cholestatic liver diseases so that we could have controls. And so our sample collection was including diagnostic and research liver biopsies, which again is where the patient voice was so important because, it's important for us to understand how to communicate the study so that patients will take that day to provide the research-only biopsy. And we collected PSC explanted tissue to look at the heterogeneity of disease as well as blood, whole serum, and PBMCs. And so our goal is to generate cellular maps to look at the heterogeneity of disease as well as active pathways that are found in early disease and then ideally, amplified in late disease. And this is work that's really been spearheaded by Diana Nakib, who's a wonderful PhD candidate in the lab, and the early work was taken on by Katya Persiani and Tallulah Andrews, who were wonderful postdocs. One is in Gilead right now, Tallulah is at University of Western Ontario, and Louis Liu, who's in medical school. And so some of the interesting collaborative efforts and knowledge translation efforts were really led by PSC Partners Canada. So I will let Mary talk about this.
Okay. Well, so this was one of our first events. This was, gosh, pre-COVID. And Sonya generously came to a meetup in Toronto and presented to about 60 patients on the work that she had done on the healthy liver and on the this study, the adult study. And interestingly enough, we had I don't know if you know this story, we had a nurse practitioner in the room who was a translator. She stood in the back of the room, and any time terminology was being used that she deemed might be difficult, she would stop and say, "Excuse me, what's a cholangiocyte?" And so we learned that having a patient translator is a really useful thing. And in fact, in the next grant that we wrote together, we included a patient expert communicator, Verva Algis, who is here at the conference. I was hoping she'd be here in the room, but I don't see her. And she is a patient who will serve in our efforts for knowledge translation to help translate some of the scientific language into language that patients will understand.
And so what we're collecting our samples for is transcriptomic mapping. So we really want to understand the individual cell populations and what genes are active in those cells at this time. There is a gap in the literature as to whether transcriptomic mapping alone is the most robust way to look at disease-specific patterns. So it is up for discussion, whether epigenomic mapping is better or whether, looking at, TCRs and BCRs is. But, at the beginning, we've been doing transcriptomic mapping. We always save samples for functional validation. So we really want to be able to, especially from our explanted tissues, we want to be able to save cells once we develop targets, and then we use the drug discovery pipeline to repurpose drugs that are already being used for autoimmune liver disease, then we can test them on these populations that we've frozen down. So we try and kind of biobank the samples stored in as many ways as possible. And then also we're saving tissue for spatial transcriptomics as well as we save cells or tissue for immunohistochemistry. And so the second study is, as Mary mentioned, is the study of PSC in children. So this was the specific call from Chan Zuckerberg Initiative, and it was a really neat application process because there wasn't, the platform was all done on Google, and everybody was kind of working on it simultaneously. So you could really see that our collaboration was not a token collaboration. Everybody was kind of writing at the same time, and you could actually measure, you track people's keystrokes. So it was a kind of a neat experience. And so this was, do you want to present this?
Yeah, well, I added this slide. What I really just wanted to point out was that in 2022, this RFA was announced, and it was entitled Patient-Partnered Collaborations for Single-Cell Analysis of Rare Inflammatory Pediatric Disease. It's a mouthful, but it's really quite interesting to have patient-partnered with single-cell analysis. And I'm not sure I can read it, but let me see. There was something I wanted to read about invites applications from collaborative teams bringing together patient-led rare disease organizations and research teams for 4-year research projects aimed at advancing our understanding of the fundamental science of rare diseases. This is really, I mean, the Canadian government did encourage in their applications to find out, they invited information about how you would include patients, but this required it. You were not eligible for this study unless you were applying in conjunction with the patient organization. And in fact, the funding is shared between the two teams. PSC Partners, when we did receive the grant, UHN received the bulk of the money, but PSC Partners also received money to fulfill the pieces of the application that we committed to do, which is primarily the knowledge translation piece. And so the title of the proposal was Understanding the Cellular Ecosystem of Primary Sclerosing Cholangitis, which got cut off. And then the next part, oh, so yes, so we've been invited, so as part of this grant, we are part of a network of other patient-partnered collaborative teams that were also grant recipients. There were 10 teams awarded grants, and this is part of a presentation that we made to that network. And this is the patient organization, myself as the patient organization principal investigator, which is also something quite unique to be a PI on a grant of this size as a patient organization representative. Rachel Gomel is named as a collaborator on this study. And then, as I said, Verva Algis is the expert patient community. And these are just some more images of us at one of these professional meetings, and then at the presentation that Sonya did for the PSC Partners Canada. And here's the...
Yeah, so, and again, the availability of these funding calls makes what we're doing in this weekend so important, because a lot of the questions that these funders have are, how can we generate data? How can we ask questions that are patient relevant? And how can we link all that data to really strong metadata and clinical follow-up? So the fact that we're, as a community, as a PSC community, kind of organizing, already collaborating, you know, it'll make it very attractive to these funders, because, you know, often you kind of have these lone wolves that are doing these individual studies, but, you know, it's not backed up with really strong metadata. And so I think what we're doing this weekend and kind of planning roadmaps and stuff like that can only be attractive to these funders. Okay, so this research team, we have principal investigators, including Gary Bader, who's a fantastic computational biologist and the liver, or like a human cell atlas kind of organizer. So he's leading the liver group with Human Cell Atlas, but he's kind of leading the larger group as well. And also Amanda Ricciuto, who is a pediatric gastroenterologist at the Hospital for Sick Children, and she's recruiting patients that have IBD and ideally are untreated for IBD, and then when they are diagnosed with PSC, again, before they're treated, we're getting liver biopsies from these patients. And then we're collaborating with Ken Koidaru, who's a very famous gastroenterologist who studies IBD, as well as Aliyah Ghulam-Hussain, Gideon Hirschfeld, who are obviously very famous hepatologists, and Ian McGilvery, Blaine Syed, who are transplant surgeons, and Ellen Mullen, a hepatologist at UMass in Boston, who is really doing a lot of heterogeneity of disease and PSC as well. And so as a group, we're really trying to transcriptomically map the temporal and spatial distribution of immune cells in pediatric PSC liver using single-cell transcriptomics. And we're comparing this to a pediatric-only healthy liver map that's been generated as part of another Chan Zuckerberg grant. So what we're trying to do is also identify shared pathogenic pathways in the gut and the liver of patients at diagnosis, and kind of match that with what's happening in our late stage samples. And so really we want to use this to generate really high quality targets and use drug discovery pipelines ideally to identify drugs that are already FDA approved and already in the pipeline and shown, you know, safety data, but to repurpose it for PSC. And so our aims for the grant is really to do 5 patients that we look very deeply at. So we want to look at matched liver and gut, and we want to look at multiple regions of the liver. We want to look transcriptomically as well as using the metagenomics, and look at BCRs and TCRs. So look very, very deeply at these patients.
So this slide is also from that presentation to the network, where we're sharing what our plans are as a patient organization for the grant activities. So the joint activities are to provide input at monthly research meetings, to support public outreach and knowledge dissemination through lay presentations such as this, interviews with researchers that we can then present to the patient community, and PSC Partner Canada organized events. We are having our first event that is funded by this project in October in Toronto, and there is plenty of room and there is travel support available. So if you are interested, please go to our website, pscpartners.ca/events. Check it out. We're very excited about this 2-day mini conference that we'll be holding in Toronto. And then centering the voice of the PSC community, expanding existing channels of PSC patient support and engagement with the research community in the US and Canada. The grant is funding the patient partner members of this team to attend professional meetings and to drive knowledge translation with a focus on DEI and accessibility. The grant is also providing funding for us to provide accessibility at the events that we're holding. We're having French translation and ASL translation available, childcare, providing travel support, and we are planning to translate our website in the next year using the support of this grant. Anticipated challenges, the diversity of languages spoken. Thus far, we have not been called upon to help recruit for this study because there's been enough patients coming through, sick kids. But should we need recruitment, we will be helping and by reaching out to our connection with the pediatric population in Canada or in Ontario. And these are just, this slide is just showing opportunities for collaboration within the network. And here's a photo of the 4 PIs at Toronto General Hospital. Do you want to say anything about your research commitments here?
Just, because both of the funders, that the commitment is not to just kind of generating papers, it's generating tools, sharing data, and providing like a rich data set for everyone to use like democratically. So the one thing that we're very much committed to, regardless of what disease you're looking at, is providing this data, open source, free publication, so that people's studies maybe it helps people's studies. So the one thing that we're happy to do, but we're also mandated to do, is to share this openly. And so one of the things we've actually found is that once you take bring people together and have everyone sharing data openly, the actual students are much happier, the research goes faster, people don't spend 6 months optimizing how to take cells out of tissue. So we're open to collaborating with anyone and everyone, and certainly providing that data through publication.
And likewise for us as a patient organization, the bullet point there that we will bring resources that we learned from this collaboration back to the rare disease community, the CZI community, the rarest one, NORD, which is the National Organization for Rare Disease.
And so just before we kind of get into the future of this work, we just want to touch on some of the data that's come from this pediatric collaboration. So we've been able to generate a pediatric specific liver map, and we certainly know from some very wonderful work on fetal liver development that the fetal liver is quite a bit different from adult liver, and what we're seeing is that the pediatric liver as well is quite a bit different from the adult liver. And some of the themes that we are seeing in pediatric liver are actually, or that in those development maps are actually seen throughout the pediatric liver. And so what we've characterized is about 30,000 pediatric liver cells from 8 samples. And what we see is that some of the myeloid populations in the pediatric liver are more pro-inflammatory. And what we're examining is whether actually pro-inflammatory is a problem, whether it's just that the immune system is becoming educated. And because pro-inflammatory systems signals have been seen in the developing liver as well. So we're using this as our comparator to our PSC maps that we're generating with Amanda. So here's our sample collection protocol. So we're collecting samples from the terminal ileum, the ascending colon, and the descending colon, as well as transverse colon. So we're collecting from 4 areas right now, and we're also taking a liver biopsy as well as blood and stool. So what we want to do is kind of do, for these 5 patients, a very rich analysis of the transcriptome as well as the metagenome to really understand what is necessary in order to, spatial transcriptomics to figure out, where is the overwhelming signal. If we had to kind of provide a suggestion to the community as to what is required, like which of these approaches are most important to see these early signals and compare them to our late signals. And we're doing this as much as possible in untreated IBD as well as untreated and at PSC at diagnosis. And about 20% of these patients then go on for resampling. So we are also poised to biopsy those patients if they have to go, or have a third pass of these liver biopsies if they go in for resampling. And so we're using Visium spatial transcriptomics for these regions, as well as we're doing single-cell RNA sequencing with BCR and TCR analysis. And so we think that is going to give us a nice look at the distribution of cells with respect to the lesions in the liver, as well as understanding the different immune populations that we're finding that are infiltrating the liver. So we've done this in 2 patients right now. We've also shared the protocols that we're using, again, through protocols.io. And if you don't have a chance to write that down, you can just email me and we'll provide it. And so we're using the workflow that we used in our bioarchive, which was for the adult liver. So we have done single-cell and spatial transcriptomics of the explanted livers in patients with PSC. And so one of the things that we're really trying to look at is giving a spatial context to the different immune populations we're seeing in PSC in our PSC maps. So what we do is we look at the scars, we map them using our pathologist maps them, and then we're looking at the individual immune populations that are either inside or outside the scars. And the reason that we're doing that is we want to understand, number one, what populations can we target, which populations might be associated with the hepatocytes that are transitioning to cholangiocytes, or different types of cholangiocytes in that lesion. And so we're really trying to map, as disease progresses, which populations could potentially be reprogrammed to provide a regenerative signal or to maybe quiet the inflammation in the cholangiocytes. And then we're also looking, again, using the 4 different regions of the gut at the different immune populations we find. So this is a bit of a busy slide, but what it allows us to do is use that platform I showed you at the beginning, the single-cell RNA sequencing platform, to see what populations are there. But we also want to look at what types of T cells are found and what types of B cells are found. So what we want to see is there a particular T cell that we're finding associated with a particular level of disease in regions of the gut, and is that found in the liver? And again, it's so that we can identify targets that we would reprogram. And so we're also looking at myeloid cells, so those are the macrophages, the kind of eaters of the liver, as well as B cells, so these are the cells that secrete antibodies and potentially autoantibodies. And so what we can see is we can capture all those populations in these different regions of the gut. But when we look at our T cells, and we do something called a T cell receptor analysis that can tell us the identity of the T cells, we're actually seeing these different regions of the gut have different types of T cells. So it allows us to start asking the question of, is there a particular T cell population that might predominate in early disease? It's also found in late disease and found in the gut and the liver. So it just gives us a rationale for sampling as many regions as specifically as possible. And then we want to ask the question about what about immune populations that are found in the gut, blood, and liver. So what we can do with this platform is really take T cells from the blood, T cells from the 4 regions of the gut that we get through the colonoscopy biopsies, as well as the T cells from the liver. And what we can do is just look at the types or the shared T cell clones. So that's the T cell receptors that are shared across these different regions. And what we see is that, the liver shares more T cell receptors with the blood, but there's also some shared T cell receptors with gut. So what we're doing is we're looking at these shared T cell receptors. What are they? What are they specific for, and how can we link that to the disease pathology? So that is some of the kind of early data we're getting from this work. And so our summary from these this early work is that we have an enrichment of unique T cells in the gut of patients with PSC and IBD. But we also see expanded T cells in the liver, the blood, and the gut that require kind of further examination. And so I'd like to acknowledge the team at Toronto. Before we kind of go on to our discussion, I just want to acknowledge that this is a huge team that's really required for coordinating. Our monthly meetings are like huge sometimes, and often it's just sometimes just a catch up, but often we're really planning the next stage. So I don't think any of the meetings ever end early, even though we may only have 2 or 3 slides to share. So it's a wonderful team, and made even more wonderful by PSC.
Well, and I'd like to say that it's a unique skill to be able to pull together a team of this size and manage it effectively, and this is something that you have experience doing, and so that's a special skill. I just wanted to share a little bit what this collaboration is going to look like moving forward. So for the next few years, well, obviously ongoing will be, and primarily ongoing will be the science, collecting the samples, processing the samples, innovating as they process the samples, analyzing, publishing, and sharing protocols and data. The monthly team meetings will, and that we will be participating with the larger collaborator group. And then for the rest of 2023, in terms of the patient part of this grant, we will be attending the CZI Rarest 1 conference, which is happening in 2 weeks, which is where it's primarily a meeting for rare disease patient organizations. And at this meeting, they've actually mandated that the scientific PI on each of these grants attend. So Sonya will be coming to my world. And then later on this summer, there will be the opposite. There will be a single cell science conference. And they've also mandated that the patient PI attend that, so I will be attending that world. So that will be very interesting for us. And then, as I mentioned, we have this knowledge translation patient event in October 2023 that we have entitled What the Cell? 2024, there will be a pediatric patient event with sick kids in Toronto, the translation of the website, as I said, and then we will be reporting jointly to CZI on our progress on this grant, and hopefully, well, we definitely will be requesting funding for the next 2 years, and hopefully we will be granted that. So this is just kind of a slide to ask if you have any questions about how might we go from this type of work to eventually something that will help us get towards treatments and a cure. Thank you. I think we have time for questions.
Should be 20 minutes for questions.
Yes. That's a solid question. That is solid. So we are separating our peds into 2 to 12, and then we're having a intermediate 13 to 18, and then we have adult, which is 18 above. And we're seeing that the 2 to 12, which is kind of around, like, the patients are usually diagnosed in that, a lot of Amanda's patients are diagnosed between like 10 and 12. That looks quite a bit different than the post-puberty pre-adult. So we're separating into like 3 groups. Yeah, it's great. Great question.
Very, very nice talk. Very much enjoyed it, and we will learn a lot about the disease, but also about the health and what we just heard about the different age groups. So that could in itself already a huge service to the research community to provide the single-cell RNA-seq data on the healthy controls. Can you share a little bit where they come from? Who does a liver biopsy on a healthy patient?
Yep, excellent question. So we have surgeons who take off the caudate lobe from neurologically deceased donors as they perform the transplant procedure. It allows them better vascular access. So for about 40 patients that we've profiled by single-cell RNA sequencing. It's from that caudate lobe. The reason that we're so excited about using the caudate lobe is that we can profile it, and then we can save the rest of those cells for functional validation. The challenge is that just by the fact that they are NDD, there is very well documented inflammation associated with being neurologically deceased donors. So these people are brain dead donors, but their liver is sufficient to be put into, to be transplanted. So they have a healthy liver, but when you look at it histologically, there's a little bit of inflammation. It isn't so much that it makes that liver not appropriate for transplantation, but it makes it not a perfectly normal liver. However, Toronto is very famous for its liver donor transplant program. So what we have is from 20 patients, we have living liver donor biopsies that we get from the family member that's donating their liver or an anonymous donor. So then we can look at the difference between our caudate lobes and then our very normal liver from a patient that is not neurologically deceased. The challenge is that from that core, we get a single core biopsy. So we can generate these maps, but we still have to validate them with liver resident cells from the caudates because we don't have enough cells from the living liver donors to look at that. Now, Diana has looked at some of the different inflammatory pathways when you compare living liver donor to the neurologically deceased donor, and it can be disaggregated. So there can be a gene signature. It's kind of like, this hepatocyte activation could be associated with neurologically deceased donor. But, it's probably our best bet in terms of healthy because, a lot of the healthy samples are, normal liver from people with colon cancer or something, a colon cancer resection. So yeah, that's our source of healthy liver. From Amanda, for our source of healthy gut, we'll have either patients that have ingested something, so they're going to have a colonoscopy for a foreign body, or it's a patient that is being examined, and then when the gastroenterologist looks through, they say this is a perfectly normal liver. So then they say, get ready, and we take the biopsies and then we process them for comparison. And this discussion of how our work will then be translated into improvements for PSC, again, is why this community is so important because, we spend a lot of time generating these targets, but we don't have the kind of bandwidth then to generate all of the models. So, depending on what populations we uncover, you may need an organoid model that's going to go for 28 days. You may need an animal model. So that's why it's important to kind of have this data, people absorbing it and looking at it through the community so we can figure out actually how to go from, say, for example, so what Gary Bader is very, very good at is he takes these maps and then uses a drug discovery pipeline to say these drugs that are already available can be used to interfere with this pathway. So it was interesting because in transplant rejection, he came up with caffeine. Like caffeine was the biggest hit, so, but the question is, once we have these hits from these maps, what is the best 3D model? What's the best animal model to test that? And so this is something that we can be doing as a community and taking responsibility for because models need to be disease specific. So, we need to have PSC specific models. And so this is something that we could lead as a community. And the other plug I want to make for patient partnerships is that probably every 3 to 4 months, Mary sends me an email and says, you have to meet this person. And inevitably, it turns into a really great conversation. So it's important for us to be thinking about our data, but it's important for also our patient partners to be thinking about our data. And Mary touched on a really great point. Like, it's important for the patient partners to be coming to the conferences and to be attending, not necessarily only patient-oriented conferences, but basic science-oriented conferences, and we have to have general sessions that are accessible to both scientists as well as people that have more of a patient-oriented view of science, right? And so having these kind of large sessions like at that Canadian liver meeting, I think is very valuable.
I agree.
Awesome. Well, thank you everybody for coming. Just a couple of reminders. At 6 p.m., there's the clinical trial landscape and PSC updates and opportunities session, which is just right next door. And then also dinner is at 7 p.m. And if you could please pick up your table cards before going to the OPM Terrace. But I think we're all set. Thank you so much, Dr. MacParland and Mary Vyas. It was such a great talk, and yeah.